Let's go to our notebook and a Jupyter Hub. Please don't make a liar out of me. There we go. Overwrite. Yeah, that's fine. Okay. I guess I didn't close that other instance when I reloaded here. Okay, cool. I'm really excited about this one too. So a lot of fishery science lives in point space. Most of the sort of type of science does, but you collect data at very specific locations and times. Trawl tows, plankton nets, CTD casts, tag detections, acoustic pings, imagery. All of it sort of anchored to a latitude, longitude, and a timestamp. The observations are very information rich, biologically speaking, but they don't tell the whole story on their own. And most of the time, especially in the fisheries world, what we really want to know is what kind of environment those organisms were experiencing when and where we observe them. So this is where satellite data becomes really powerful, and also when things can start to get a little messy. Pulling a single pixel from a single satellite image is usually pretty straightforward, as we'll demonstrate. But once you start dealing with a lot of points spread across different days, maybe even years, the process can get slow and very painful very quickly if you're doing it by hand. So in this tutorial, we're going to kind of build a workflow that helps automate that matching step. So lining up the fisheries observations with the right satellite data in space and time. We'll take it one piece at a time so the logic stays clear and reusable, and then we'll have some fun and do some science along the way as we're kind of going through. So instead of framing this in kind of hypothetical space, we thought it might be nice to fiddle around with a real fisheries data set. So we're going to be using data collected by one of the NOAA fisheries biannual surveys collected in the Gulf, specifically from the Southeast Area Monitoring and Assessment Program, or CMAP. So if you have not worked with CMAP before, it is a gold mine. Species composition and size and abundance and environmental measurements, all focused on the U.S. Gulf Coast. It's a great sandbox for experimenting with these kind of integrations. So by the end of the tutorial, there's four main things I want you to be comfortable with. First is running your own sort of run-of-the-mill pixel extraction from a satellite image. Then we're going to import real ship survey data and visualize those sampling locations directly on top of the satellite image. Then we'll extract the pace reflectance data. Yes, I'm still forcing to work in reflectance space at each of those appropriate locations and dates for multiple observations at once, not just a single point. And then we'll use those combined data sets. We have it all together. We extract a bunch of reflectance data at these timestamps. Why not take a stab at building kind of a simple proof of concept model that tries to relate fish abundance to our satellite-derived variables? Nothing fancy. The goal is just kind of to understand the workflow, not necessarily to build a production-ready model. All right, so let's start with this sort of simple exercise here. We're going to extract pace reflectance data from a single defined location here on a single date. So feel free to change that date and location below. I did pull an example that was at least relatively cloud free, so we can get some nice results here. And then we're going to use our friend Cartopy, yeah, let's just view where this is at. We're not in satellite space right now. All right, we pulled that little dot straight from kind of Chandelier Islands region, which I love. It's a little fascinating estuarine region. Great shark nursery. Probably find some sawfish there. Anyways, let's grab a pace image from this day, and we're going to place our predefined time variable in the temporal section of the query to NASA. So we're going to time. Now we defined up here. Let's grab that. Oh, hold on. Hey, see my memory filling up. I still have the other one loaded. When that goes, I'm going to go ahead and restart my kernel. I'm glad you all saw this, because you may have the same thing as well. So we're going to restart kernel. Just going to bring that memory back down. don't know if that is true or not. Definitely restarted kernel. Let us, just for kicks, let's go ahead and just close this instance and make sure, because we don't want to crash everything. Tutorials, matchups. Let's go back in here. Yeah, all right. Quick, tell me I'm running out of memory. Hopefully I don't have to completely restart my instance here. All right. We'll see what happens. Let's see if it's just hallucinating on us.
Yeah. Ryan, did you try shutting down all kernels?
No, shutting down all kernels. All right, let's try that. Ah,
Yeah, see,
5
it's got
running
5
kernels.
of them.
Yeah, there you go.
That should do it. Yeah.
There we go. All right. But I do need to restart it. I'm going to shut it down. There we go. All right. All right. It's wonderful when things like that happen in the tutorial, so we can show you all what to do when that happens. All right. Let's go ahead and load up our data here. I'm going to give a little sidebar here as we're loading this up. You'll notice that I've chosen to get level 3 data here. Before we go any further, I kind of want to pause for a second and talk about the satellite data level we're using, because some of you who work closely with ocean color imagery might already be cringing and raising an eyebrow. Why are you telling these people to use level 3 data? Stay in level 2, right? If your background is in biooptical algorithm development or sort of pixel level validation, your instinct is right. Level 2 is usually what you want. The level 2 preserves the highest spatial and temporal detail. They're absolutely critical if you're trying to kind of reproduce or evaluate a biogeochemical retrieval at a specific time and place. Fisheries is different. Their questions tend to live at different scales. Most of the time, we're not asking what was the exact chlorophyll concentration at this pixel at 10:37 a.m. We're asking something more like, what kind of environment was this fish using? Because they might be moving pretty far over the course of a day. Fish are not passive tracers. They're moving. They respond to gradients and features and conditions that integrate over time and space. So the value of a single sort of variable at a single pixel at a single instance is rarely the whole story. What matters more are the things like recent productivity, the water mass structure, persistent fronts, or recurring habitat features. So that's where level 3 can be a really good fit. Level 3 products, they kind of smooth and aggregate the signal in such a way that reduces the noise. It'll improve your coverage potentially if you're using composites, and it'll better reflect the environmental context that mobile organisms are experiencing. So we are intentionally giving up some fine scale precision kind of in exchange for stability and ecological relevance. So, and to be clear, we're not doing algorithm development here. We're not trying to validate satellite retrievals. We're trying to build ecological models. And in that setting, accepting some spatial and temporal averaging isn't a bad thing. It's a working assumption that's probably closer to the biology of the system that we're studying. So thank you for coming to my TED Talk on the matter. So let's go back to our pixel. All right, we predefined that lat long earlier. So yeah, we predefined that lat long. So we're just going to extract a point. We'll put this little time thing in here just to see how long it takes. And that's it. We're plotting up a line. 2 lines of code here. Bam. All right, we took a spectrum at that single location. That's great. It's the same brown, super brownish water. Again, that extremely low UV reflectance tells us that there's a lot of CDOM in the water, which absorbs UV light like nobody's business. There's also a lot of phytoplankton galore here. This little bump centered around kind of the 680 range here. This is what we're measuring when we're looking at the fluorescence line height, sort of peak here. Phytoplankton absorbs sunlight to photosynthesize, but not all of that energy is getting used. Some of it's, again, being re-emitted as this sort of faint red glow, and it's essentially excess light they did use, and this kind of gives us a direct optical signal of living phytoplankton in the water, you know, when you have this. So, all right, let's throw a few points at it. We're gonna loop through these 3 points. Let's just define 3 points instead. Bada bing, you know, near instantaneously, again, we get a return. It's pretty straightforward, and it takes very little code to be able to do that. But let's just add one simple addition. Let's do the same thing, but over multiple days, like a lot of days. Because if you're working with a long-term data set like MODIS and you've got 20 years of trawl data, you might be extracting thousands of points from hundreds of days. So now we're going to pull out our sort of real world scenario here. So like I mentioned up front, we're going to work with some real ship-based survey data from NOAA fisheries coming from CMAP. This is data I pulled from 2024 onward. The idea here is pretty simple. For each survey station, we're just going to reach into the satellite record and pull out the pace remote sensing reflectance at those same locations and dates. Okay, so we have a location, date, and we have decimal, latitude, longitude. I think it might be starting and ending or something like that. So yeah, we have. I think we averaged it. Anyways, yeah, that's what we're pulling from. Oh, there we go. Latitude and longitude. That was averaging the starting, ending latitude, starting and ending longitude. So we have a latitude, longitude that we'll pull from. Lat, long, location, date. This gives us a clean way to just pair point-based biological observations with spatially continuous, satellite data. We're going to focus on the workflow here. So we're starting with this pre-clean CSV. Has all this stuff. You also notice that it has things like temperature, salinity, and oxygen. Look at all these really cool data sets. You know, it has chlorophyll from fluorometry, turbidity, Secchi depth, Férot-Houlet scale. It's super cool. But we want some kind of fisheries response variable. So in this case, I'm using this tote live. I had to kind of really dig through the README here, try to figure out and interpret all this stuff. What tote live is, it's the total wet weight of everything that was caught in tow. Fish, crustaceans, whatever else came up in the net. It's not really the most nuanced fisheries metric, but I think it's fine for demonstrating how this kind of satellite survey matchup works. And the nice thing about CMAP is that if you want to go deeper later, there's a lot more under the hood. There's species level abundances, size, data, all sorts of stuff. So if you do want to, and we could pull any one of those out here by just, we define track DF as this read CSV, and it's really easy to just pull individual variables out, which we'll use later as well. So yeah, if you do want to follow along with, later, if you want to just plug in your own, your own data, if you just rename your column of date, _ date, this should work seamlessly, and latitude and longitude are there. It'll read it in. If not, all you have to do is just change some things in the code just to make sure that it's reading in the right variable. But it might be easier just to change your your thing there. But all right, so using pandas to kind of read in the CSV file, it's heaven sent. So it just makes it a lot easier to kind of interact with the data. All right. Yeah. So the other thing, what we're doing here, if we're on any given day, they sampled more than one station, right? So if we're not careful, what we could end up doing is pinging NASA for data for every single 594 lines in the spreadsheet. We want to be smart about this up front. So let's sort the dates, and then we're going to isolate only those unique dates from there. So we have 61 unique days in there. That's going to save us instead of going through, when you're kind of cloud computing here, every time you try to pull a pixel or do some stuff like that, it just keeps pinging, right? So you don't want to do that 500 times. It's really going to slow you down. Okay, so we've got the 61 unique dates. So let's get a visual here of where all these locations are across all these 61 dates, just so we get kind of a geographic extent. This is from 2024 on. This is an awesome data set here. So we kind of subset this data as well. Level 3, again, we're reading in an entire global file. All right, so we're using this slice function. We define the latitude limits and longitude limits, and we're just Now, when we start pulling data, we're just pulling it from a subset of the Gulf here. And then we pulled out one of the wavelengths, remote sensing reflectance. You can use the nearest function to go ahead and pull the nearest wavelength to 443, which actually ends up being 442 here. So this is useful if you want to pull individual wavelengths that you want to check out, and then you can go ahead and plot those up. So my screen keeps resetting here. Okay. Yeah, I kind of like to go through this just so it wasn't intuitive. You know, we have this fig ax option. Kind of the easiest way to think about this is like figure is the whole canvas, and then ax is the actual plotting surface where data gets drawn. Going over this because it drove me nuts, and I was trying to figure this out, you know, with Python and converting over from MATLAB. So almost everything you see on the map, the image, coastline, points, labels, it's being added to that same axis object, AX. When we specify a projection here, in this case, plate Curie, we're saying that this axis understands latitude and longitude. That's important because everything we add later needs to sort of line up geographically. So this is what allows us to layer different information like coastlines and our data points on top of the satellite image and then have it all sort of align correctly. Again, it might seem nitpicky to kind of point out these little things, but it's easy to lose sight of the logic if you're kind of following along, especially if you're new to Python or coding in general. So, all right, now these data points. Before we extract our data, we have a decision to make here. What scale do you want to extract your satellite data at? Get ready for another just tiny TED talk here. The right answer really depends on your question. If you're looking at broad habitat use for highly mobile species, a shark or tuna, grabbing the nearest 8-day composite might be fine. Most of the features they might be responding to might be sort of larger mesoscale features. They're persistent on sort of weekly scales anyway. But if you're working with things like plankton tow or shelf species that might be more tightly coupled to short-term processes, daily data might make more sense. The trade-off is cloud cover. Daily imagery drops down your sample size fast. In our case, we're switching from an 8-day to a 1-day composite. Takes us from about 500 matchups, so almost our whole data set, down to 200. So if your science question can tolerate a bit of temporal smoothing, longer composites can really help with coverage. Just be clear about the assumptions that you're making. Here, we're not tracking tuna across the Pacific. This is trawl data on the continental shelf where things like river plumes and mixing can change very quickly. So for that reason, I'd probably keep the temporal window tighter in this case. Now, what if I ask a different question? I'm not trying to look at abundance in this. What if I'm asking how these species maybe have changed over the course of time with sort of differences in the location of the loop current eddy that comes up, right? We have this giant eddy that comes up. Maybe I'm kind of interested in how some of these species along here might be responding to that. In that case, if I'm sort of looking at climate scale issues and looking at differences from year to year, I might take monthly composites in that case. Just think about the question that you're asking, and sort of the temporal scale can really, it might sort of define on what you're asking and what sort of processes you're looking at. If you're looking at long-term climate signals, you kind of want to see how, in general, this whole region is changing, not just very specifically. Because if you're very specific about that, you're gonna get caught in the basic variability of noise and not the climate signal, right? So, okay. High level of what we're going to do next is basically a single matchup to many matchups in a way that's efficient and memory safe. So for every unique sampling date in our CSV, we query NASA for the corresponding level 3 PACE data file. And once we have that file, we loop through all the survey points collected on that day, and then we extract the nearest satellite pixel for each one. rather than writing to the disk over and over and over again, we store everything temporarily in a dictionary. And then we collect those dictionaries in a list and then append everything back onto the CSV in one shot at the end. So this keeps things kind of fast and tidy. One important implementation detail here is memory management. We only ever open one satellite file at a time. The Earth Access Open, calls the stream. It streams the data, so nothing gets fully loaded into memory unless we actually touch it with Xarray. On top of that, we have this with statement here. With that with statement, just make sure that each data set is closed immediately after we're done with it. So once that block ends, the memory is released, and just helps keep your kernel alive, especially if you're working in the cloud here. So this day-by-day approach though is kind of the main bottleneck. If you have a lot of local memory, it can be faster just to download everything first and then just process it all at once. The processing and extracting will be lightning fast. Here we're choosing stability and kind of portability over raw speed. For this example, I'm not going to run it right now because it does take a few minutes here. Feel free to skip running this section if you want entirely, because we're gonna extract what we read out here. We have this kind of hiding in the supporting files already, so we can extract that. But you can look at the output in the supporting files and see what it looks like. Anyways, so the main part is kind of we're using the same function we used earlier, using Xarray. We're going to extract the nearest pixel here using the nearest, right? We're looping over our predefined lat longs, just like we did earlier, except we're taking this from the spreadsheet, matched to a specific date, and looping over multiple dates. So we collect this in output kind of in a dictionary, and then we write it all to a CSV. In a lot of satellite workflows, instead of grabbing just a single pixel, it might be common to average over a small neighborhood, like 3x3 or 5x5 pixels. It's generally more common with like the level 2 files, but just kind of gives you an idea of the variability. This does smooth out the noise. It also reduces the impact of clouds or bad pixels. Instead of grabbing that central one, you might have a larger chance of getting more data if you're averaging over a larger area. So even though the level 3 products are already binned, the same idea still applies, right? For habitat questions, perhaps a little averaging can kind of better reflect the environment a fish actually experiences, since they might be moving over that distance over the course of a day, rather than the value at one exact grid cell. So below, this is the same exact thing. It just shows you how to adapt our previous extraction to compute a simple 3x3 mean around each survey location. This is where the meat happens here. If you want to change this to an 8-day, just change this to 8D right here, and you'll extract the 8 days instead. One kind of nuance to this, there's a way to do it. It's kind of a little complicated, and I didn't want to mess with it here in this tutorial. But if it picks a date, it's just going to take the 8-day that it overlaps with. But if it's on like the first day of that 8-day file, sometimes you might be able to extract the nearest sort of middle date. I don't know. I think it's a little too nuanced. But if you put 8D in here, where that date intersects with that 8-day window, it will pull that specific 8-day. So you're probably going to be pulling a lot of the same images over and over again when you go to consecutive days. But just so you know how to work it, how to run it. So maybe a little more detailed. It's just a small tweak from kind of the center pixel extraction. When you use, there's something important to note here. We used this nearest earlier, right? Where did we do that? This method nearest to do that. It gives you the data value at the closest coordinate, but it doesn't tell you the actual index position in the array. So if you want to go back and grab a block of pixels around that point, you need the indices of that location to kind of slice the array. We're using the slice function, right? So you need those indices of those locations. So that's why I use this NP, absolute value of the array minus the value. We're finding the arg min. We're just manually computing, all right, what's the closest latitude and longitude, for our given location that we're feeding it. So it gives it the numeric index directly. Just makes it easier to define this 3x3 window around the point. This may be completely unnecessary, probably use nearest still. This is how my mind kind of logically landed here. And there might be quicker and fancier ways to do that. And I am very open to hearing those, because this does take a few minutes to run still. All of my talking, it could have run this whole time in the background. But, okay, so we run through that. Let's, we'll assume that it went through and we've matched each survey data point to a satellite spectrum. So let's take a quick look at the data here. Let's see, we're loading it in. Again, this read CSV is just great. This pandas thing makes it so easy. Again, for simplicity, we're just going to focus on this total live variable in the CMAP data, which represents that total catch in kilograms. For habitat modeling, we don't really expect a super tight correlation between remote sensing reflectance and total catch, but it's useful to explore how including PACE data might help. So first, we open the CSV here. We get a sense of the average reflectance shape across different biomass levels. If we print the min and max of this data, we see that it spans 3 orders of magnitude, and we're talking biological data. So that immediately tips me off. We're probably going to want to work in the log space here. If you take the log of this data, it kind of normalizes the distribution. So when you apply parametric statistics to the data later, you don't have these extreme outliers that are driving your correlations. You're like, this correlation looks great, and it's just because of these like 2 data points way off in the distance here. So what we'll do below is we'll transform the data by taking the log 10, and since it's more evenly spaced, and then what we could do is we'll divide it into 10 equivalent bins after we transform it. So we're kind of making things linear in log space. Then we're going to take 10 bins, and within each bin, we're going to take an average of all the reflectance data within each little bin, and let's just see what it looks like. That's what we're doing here. So yeah, we're binning in the log space and taking the mean spectra of the reflectance within that. And then we get this output here. So you can see the various bins. We divided it into 10, but not all of those. You can see there's a gap here. Not all of those bins were represented in the data set, so it's just skipped over them. But basically, this is the range of kilograms that were caught. And within that range, this is what the reflected satellite, the satellite reflectance looked like. In general, we see higher catch in greener sort of phytoplankton-rich waters and lower catch in blue waters, kind of, and that. It's not too surprising, but there's this really interesting exception here. It's this lowest biomass bin. It's pretty green shifted, and a lot of absorption going on here. The reflectance is much lower. It looks different from everything else. This likely points to strongly absorbing material, maybe a CDOM plume from river runoff. If this were the case, it's also likely to show up as lower salinity. But it still doesn't explain the low biomass. So let's take a look at other environmental parameters from these same biomass bins: surface temperature, surface and bottom salinity, and bottom oxygen as well. So let's go ahead and take those out. Grab and temp surf, sal max, oxy. The max is the maximum depth. So and then we're just gonna go ahead and take the mean of those within each of those bins here, right? And those bins are organized top to bottom here. Let's see. So this is kind of that little aha moment here. There's a big difference between the surface and bottom salinity here in this first bin here, showing that there's this freshwater lens that has stratified the water column. Sorry, this first bin is actually the second one. Or 3. Here we go. So yeah, there's still this huge difference in salinity here, right? So we have a stratified water column with high nutrients and low light at the bottom. We have trapped water. Oxygen is just getting depleted by microbial respiration, and it's not getting, it's not having any more oxygen being introduced into that system, because no photosynthesis can take place because it's just so darn turbid, as we saw from that reflectance spectra, right? So now we have hypoxic waters Check out that oxygen concentration, almost near zero. The previous one's a borderline anoxic here. No wonder why there's no fish here, right? Anyways, we're veering off topic a little bit, but takeaway here is we're just looking at these reflectance patterns. It can kind of flag areas with unusual environmental conditions, or just kind of pull this out, a way to just kind of just high level try to think about what's happening in our data. This is nothing but data exploration. Like that, this plot is probably wouldn't go into a paper. This is just us trying to kind of figure out what the heck is going on, get a first order look, and really get comfortable with the reflectance data and try to figure it out, because it can give you a leg up. If you're looking at chlorophyll, it would not have tipped you off to this at all. It just would have been a high chlorophyll value, would have just blended in with everything else. So, all right, do something else fun. I like correlation matrices. There's a recent paper that Heidi Dearson had put out, and she's working with PACE data and showing, hyperspectral, all the wonders of hyperspectral data. Now we can start kind of teasing out which spectral features might relate to fish catch here, even if we don't expect a strong relationship. So we're going to use that tope live again as our target here, and compute a simple 2-band index here. Let's take our reflectance out here from our previous area, and I'm grabbing everything between 400 to 700 nanometers, just because the data outside of that is a little noisy. So let's just stick to the wavelengths that we know are really strong here, just to avoid having artificial things pop up. So we're going to compute a simple 2-band index for every possible pair of bands. So here we're using a normalized difference index, or NDI, because it's easy and it's really well behaved. You can use any other combination to, it's just NDI is really convenient for this demo. So for each band pair, we calculate the normalized difference index and then correlate it with our log transformed fish biomass. So plotting all those correlations on a heat map basically just it quickly tells us which band combinations look most promising. We have more than 100 bands from PACE that we're throwing at this, so we can kind of explore spectral relationships that haven't been tested before. So let's give that a quick spin. Might take a few seconds here. A few quick notes on the code. The kind of double loop here provides the every possible 2-band NDI, and then correlates it with our target variable here, right there. We use the sort of NP tri U here just to mask the upper triangle of the matrix, so we're not plotting duplicate correlations. It's symmetric, but just kind of helps with visualization. And the NDI correlation target, that's where we're actually calculating the Pearson correlation between the NDI and fish biomass for that band pair. Everything else is just formatting the heat map kind of nicely. All right, so basically the deeper red and the deeper blue areas are showing us which wavelength pairs show the tightest linear relationship between NDI and that log transformed total biomass. We're looking at linear. We did log transform, but we're still kind of looking at linear in space here. But we are using, even if we're looking at a 2-band ratio, we're still using all that hyperspectral information to find the best pair. But this is still pretty qualitative at this point. So we can use this matrix to kind of, explicitly pull out the band pairs that have, let's say, the top 5 highest correlations, and because we're hyperspectral, we have a lot of cross correlation. It's no surprise, because the top 5 are essentially varying versions of the same exact band combo. They're just all kind of right next to each other. You could expand this more if you do this order index to 10 or 100 or what have you. You can see what those when it starts to change and kind of go away from this. But for a while, it looks like this is our preferred band combination here, and here's your Pearson correlation coefficient. So will PACE actually do anything here? Is it going to improve the basic fish abundance model? Probably not a silver bullet, but our top correlations aren't terrible. All right, that's not horrible. So let's see what happened when we combine the most promising spectral feature with the in situ measurements we collected on the ship here: temperature, salinity, oxygen, chlorophyll, turbidity. And we'll use a tool that's starting to become more popular. I'm seeing it more in species distribution modeling, boosted regression tree. So this method builds a lot of basically small decision trees, each one learning from the error of the previous, so kind of capture nonlinear relationships and interactions without us sort of specifying them in advance. So we're kind of moving away from that linear space. The linear space is just tipping us off. Where do things tend to be sort of correlating? Now we're gonna let the nonlinearity portion let it fly. We'll also use a train test split that Sachi had mentioned earlier. So we fit the model based on one subset of data and then evaluate it on another. Ideally, you have a completely independent data set to do that. Not always, you don't, that's not always the case. So here's one way you can at least sort of evaluate your data just by holding 20% of it back. And it helps prevent overfitting and give a bit more of a realistic sense of performance on new data. But we are dealing with a relatively small data set here, so overfitting is probably inherent to this. But for demonstration purposes, I think that's it's okay. So we'll put our winning two-band combination here, and then we'll grab some other variables from our spreadsheet. These will be all our predictors, which we call X down here, our predictors, and so the RRS ratio plus our environmental variables. That's what we're trying to plug in, and then Y is the tote live log transform. That's what we're trying to predict. Not all stations had all variables sampled. Kind of frustrating. Even if they were, and you had chlorophyll and oxygen and temperature all sampled, it doesn't guarantee that there weren't clouds obscuring the satellite. Machine learning is great and very powerful, but if you want a really rich data set to really get the best use out of it. In this case, the total number of instances where we have all 6 variables co-collected has dwindled down to 97. So just saying, take these results with a grain of salt, and let's just focus on the workflow here. But you can kind of see, for these type of things, you can interpolate in between. I don't necessarily recommend that, but if whatever your predictors are, you want to make sure they are all co-occurring for this to work well. So actually, I think it'll choke up if you don't. All right, so we split the data into training and test sets, 80% for the subset, the 80% for the training, and then 20% for testing. And then we set this random seed for reproducibility here. This just randomly selects the data. The 42 is just the answer to the life in the universe and everything. It's completely nominal here. So it selects the data, but only selects it once. So if you run this again, it'll look the same. So I wanted to do that for the tutorial, so I don't run this and I'm like, hey, this looks cool, and then whatever. But if you want to randomly select data each time, remove the random state portion here. You might want that if you want to do sort of Monte Carlo analysis and say, let's run this 1,000 times, and then we'll take the average of that. That could also kind of help with reducing overfitting, and then you have an idea of uncertainty around that as well. So next we'll run we'll fit a boosted regression tree here. It's dangerously easy. Let's just build small decision trees sequentially, each one learning from the errors of the next. And parameters like a small learning rate and subsampling can help reduce overfitting as well. If you set the learning rate really high, then yeah, you might get a better answer, but you run the risk of overfitting here. So once trained here, then we predict this held out test data. We held out 20% of that, and then we're going to evaluate the performance using correlation, R squared, RMSE, and the median absolute percent error. I left these in the log space to kind of tell you how well the fit is, and then I ran these in linear space and back transformed them here because otherwise you can't get kilograms and percent out of this, and I just want it to be somewhat interpretable so we have some idea. So just FYI. All right, so to visualize the results here, so yeah, you can see how yeah, 0.709, that we're moving up in the world here. That's not too bad. So allowing those linear relationships really kind of help things. That's not bad at all, right? So to visualize this, we have two plots here. We're going to do a scatter plot of the observed versus the predicted values, kind of basic scatter plot, and then we're going to use a star sort of polar plot, which shows the relative feature importance, which is really, I think it's just a great way. You can use it as do it as like a little histogram too, but I like the star plots. I just think they're really attractive here. So, in this model, surface temperature looks like it had the biggest influence, but this PACE NDI came in a very close second here. So, and this captured a pretty nice directional trend. Again, we subset this, and when we took 20% of the data of those 97 points, we're only running this on 20 points in the data set. So it's I wouldn't call this, as we ran this, a super robust predictor. But interestingly, if you remove the PACE data, you do get more observations because cloud cover is an issue where we're just working with just the ship data. But the model's predictive relationship weakens, showing that just even a small amount of hyperspectral information can add some value, even with limited samples here. I mean, if you want to test that, I think we would do that here by just blocking out the RRS ratio. I don't want to mess up that whole line. Let's get confused between the comment outlines. No hashtag here. Let's take the RRS ratio off, and I think it'll run like this. Let's see. All right, that's our predictor is used. Oh, 256 samples. That's great. So yeah, it went way up. Okay, we're going to do the same thing. Yada yada. You can see our correlation went down. So just to kind of give you an idea. Again, the temperature ended up being kind of the most important. I'm still kind of trying to get used to how to interpret these. The relative influence of turbidity now is probably higher than it was before, because there might have been some cross correlation between that difference index and the turbidity. That some of that cross correlation is trying to be worked out in the boosted regression tree here. So, anyways. Now the science begins here. So the tutorial here kind of just highlights a workflow that's common in satellite data applications, right? Where we're just matching observations to environmental data, exploring relationships, and just kind of trying out really simple predictive models. It's not about the exact numbers; it's just about the approach. So we covered handling satellite data matchups, extracting data. We're starting to explore how environmental and spectral variables relate to these biological patterns. So with more data tuning, these methods could definitely be extended to study habitat use, fishing hotspots, other ecological questions. So hopefully this kind of gives you a solid foundation for kind of integrating remote sensing into your own fisheries workflow here. So now we'll stop there. All right, I think we're right up against time here. So if anybody has a quick question, welcome to. I don't know what comes next here. Oh, we have a half-hour break. Yes, this is like our lunch break, I guess, right? So feel free to go on if you want to hang back. I'm happy to answer questions. Let's see. Let's see if I can go back on. All right, there's one here. Ryan, you mentioned that for fisheries, L3 data is often more suitable than L2 data because they're interested in the overall environment. Organisms develop rather than pinpoint temporal and spatial accuracy. Following that logic, why is it still standard practice to associate fish catch with chlorophyll measured at the exact same instance? If a predator is the result of trophic succession, zooplankton small fish that started from a bloom weeks or months ago, shouldn't we be looking at the history of that environment rather than its current snapshot? Love that question. Yes, right? I mean, again, it depends on the question you're asking. when we're looking at remote sensing reflectance data, depending on how you want to define environment, yes, chlorophyll is food. Yes, chlorophyll is what fed those zooplankton that the fish fed on. And then, there's a lag of weeks associated with that. Or, when there's an adult catch, years, right? But they're also responding in real time to other aspects of the environment. Chris Hake, I'm so excited for you guys to hear this on Thursday. He's going to give a great talk about putting different models, some optical parameters into fisheries species distribution models. And one of the things he found is like vision is a really important aspect of fish behavior. They use it for foraging, predator avoidance, or catching food. There's still aspects of the optical environment that don't involve food. So it's not necessarily just about chlorophyll. You're still kind of characterizing, I guess, that sort of environment in that way. But if you are interested then in chlorophyll, then the question kind of becomes different. Kim, please chime in.
Yeah, I would say another aspect is at sort of pinpoint locations is it could be an indicator of a hotspot where you have fishing aggregation. If you have increased chlorophyll due to, say, localized upwelling, we're looking at that right now where we have large aggregations of whales and other fish, et cetera, in one area. Well, what's going on? Something's driving that. So that's where some of that pinpoint matching up with some of the data is also helpful. But I agree, there's both aspects of what are the long-term trends and changes. Is it a mismatch with the spring bloom? So there's different aspects of how to go about it. And some of that is then deriving indicators from this other data as well. So different ways of looking at it for sure.